Last time on Science in Real Life, Joyce, Carrie, and I went on a botanical adventure. We collected and preserved plants according to methods used by botanists for centuries. And now it's time to show you why.
The plant we collected in the last episode is still drying, so we're going to prepare a specimen to add to the herbarium that's already dried. A herbarium is a research collection of pressed dried plants. The University Herbarium here at UC Berkeley was established in 1895 and contains 2.2 million plant specimens from around the world. The Jepson Herbarium was established here in 1950 specifically to research and conserve the plants of California. So let's get to work creating our herbarium specimen so that it can be used by researchers for hundreds of years to come.
The first step is to use glue to mount our collected plant onto an herbarium sheet, which is made out of special archival paper that will help preserve the plant. That's why it's so important that we press the plant in a way that showed all of the useful characteristics, like the top and bottom of the leaf. The specimen is then labeled with all of the info that we recorded in the field so that we can preserve that data forever.
Once the herbarium sheet is complete, it's filed by scientific name and geography. Since the goal is for these specimens to be preserved forever, herbaria are climate controlled and the cabinets are routinely treated with insecticides.
Okay, I think it's time to geek out about all the amazing ways herbaria are used for research.
Yeah. Every time a new species of plant is discovered, something called a type specimen is deposited in an herbarium. The type specimen serves as a standard to which all other plants that might be that species are compared. Type collections are extremely important and are usually filed separately from the rest of the plants. Berkeley's herbaria contain around 14,000 type specimens.
Okay, but if the goal is to collect all of the information about a specimen when it's collected, why do some of these herbarium sheets have new labels that don't match the originals?
Well, herbarium specimens are not meant to chill in cabinets for hundreds of years. They're dynamic collections, so when every time a new discovery is made or plant families are renamed, experts in those fields have to go back to the original specimen and revise the labels based on current knowledge.
So it's like editing Wikipedia.
Yeah, but you have to be an expert.
We want herbarium specimens to last forever, but it's totally cool to take small tissue samples from them for research purposes. That's part of what they're there for. One example is sampling isotopes and chemicals to assess pollution levels and climate changes over time. So I could sample all of the grass specimens from San Francisco over time to see if pollution levels have increased. One of the huge advantages of preserving plants in herbaria is that their DNA is preserved too. We can sample plants that are 100 years old and still get sequence data from them. So if we needed the DNA of a rare plant from a remote jungle, we just have to stroll down to the herbarium and take a sample from the preserved specimen. It's amazing.
I know, when herbaria were first started, we didn't even know what DNA was. Now we have decades of genetic data to draw from. So we're constantly coming up with new ways to use herbarium collections as our technology advances. We can get DNA samples out of plants that are extinct, something that would be literally impossible if plant libraries like this one hadn't been created when they were.
The sheer amount of data in this herbarium is staggering. There are millions of specimens, and because we can sequence the DNA of so many of them, the herbarium contains their entire genomes. That's a quantity of data on a scale that you won't find in other places.
Many herbaria have begun digitizing their collections by scanning the herbarium sheet and creating databases of associated collection information. Scientists can now access data from herbaria across the globe for large-scale analyses. For example, they could mine the databases for collection date and location of a certain rare orchid species to understand how its global distribution has changed over the last 100 years.
All three of us use herbarium collections in our research. My first research experience ever was in the herbarium of the New York Botanical Garden. I was studying 3 super closely related sedge plants, and I took all of the specimens the herbarium had and measured their reproductive parts to see if they were different enough to justify calling them separate species. And then the next summer, we sampled them all for DNA to see if sequence data could help us figure out their evolutionary relationships.
Like Molly, my first research experience was also in a herbarium at the Smithsonian Museum of Natural History. I went through hundreds of herbarium sheets, characterizing the itty-bitty veins of the leaves, searching for unique aspects that only closely related species shared. I continued to work in herbaria through undergrad and now. All of my collections of vines from fieldwork will eventually end up here at Berkeley in these collections.
I study the climate where plants with tubers, bulbs, and rhizomes grow. I use data from tens of thousands of digitized herbarium records from around the world to determine the geographic ranges of those species and what the climate is like in those regions.
The breadth and depth of specimen diversity at herbaria like UC Berkeley's is incredible, and it's really awe-inspiring to think of the centuries of research that plant scientists have produced by making use of herbarium collections. And starting an herbarium collection is totally something you can do yourself, even without the fancy cabinets and drying equipment. Follow our how-to about plant collecting and pressing from part 1 of this episode and you're good to go. Start by getting familiar with the plants in your own backyard and see where that takes you. Carrie and Joyce, thank you so much for botanizing with me the last couple days. It's been so much fun.
Thanks for coming.
It's been great.
Don't forget to subscribe to Science in Your Life, and if you'd like to know more about Joyce and Carrie's research, check out the link to the Specht Lab website in the description. To find out more about the university and Jepson Herbaria at UC Berkeley, the link to their website is in the description as well. See you next time. Was brought to you by the American Society of Plant Biologists.